Bioblitz, reel number 2. Ever wondered why we age? And does it have anything to do with telomeres? Let's find out today in this video. One day, you're a baby, and many years of cell divisions later, you're an adult. However, eventually you and your cells start to age. Why does this happen? One of the many reasons is that over time our cells lose regenerative properties, meaning they simply stop dividing. To understand this, we must first learn about telomeres and their role in our cells. Telomeres are repetitive non-coding nucleotide sequences, which means that they do not code for any protein. They are found at the ends of each chromosome. And so the question is, what do they actually do there? They solve a very serious problem. Let's see how. When DNA polymerase replicates our linear DNA, it doesn't fully copy the ends. Hence, if there were no telomeres in transferring the DNA to our offspring, important genetic information which codes for our characteristics would be lost. Telomeres are thus referred to as protective gaps of the chromosome. However, as young cells divide, the length of telomeres also becomes shorter progressively. The cells with short telomeres are referred to as aging cells. This is what the chromosome of a young cell looks like, whereas the chromosome of aging cells has lost a significant portion of its telomeres. When the length of the telomeres becomes alarmingly short, the cell may either exit the cell cycle and stop dividing or carry out apoptosis, which is programmed cell death, so that it doesn't transfer damaged DNA to the offspring. But what about cells that must undergo repeated cell divisions? Stem cells undergo many cycles of division to produce more stem cells, as well as differentiated cells, and the same is true for germ cells, which increase their number through mitosis before undergoing meiosis to produce gametes, which are sperm and egg cells. Well, don't worry, our cells have it covered. Normally, after repeated divisions, the telomeres would have reduced greatly. However, an enzyme called telomerase prevents the loss of telomeres by adding repetitive nucleotide sequences to the ends of telomeres, thus maintaining their length.